Hey everyone, welcome back to Cell Biology. We are in the middle of talking about how different proteins are targeted to different components within the cell. We talked about different kinds of tags that can either keep something in the endoplasmic reticulum or allow it to be sent out and retrieved back. And we talked about different tagging that allows something to be sent to like mitochondria or peroxisomes. There's also other kinds of ways that we can signal proteins for where their location is. Some proteins that are going to be used in the Golgi complex can have retrieval tags or retention tags that either retain them within the Golgi or when they leave the Golgi, send them right back and retrieve them. Large complexes can be excluded from transport vesicles, and that can be involved in maintaining protein composition of the Golgi. And third, the hydrophobic regions of the Golgi can actually be involved in keeping proteins within they need to be because of the membrane spanning domains of those different proteins. So all Golgi-specific proteins are integral membrane proteins. All of the Golgi-specific proteins will have one or more membrane spanning domains. And how long that hydrophobic domain is could help determine which cisternae each protein is incorporated in, because the thickness of the cell membrane increases from the ER to the plasma membrane. Here at the ER, the length of, or the depth of the plasma membrane, of the endoplasmic reticulum membrane, it's around 5 nanometers. By the time you get to the plasma membrane, it's around 8 nanometers. So as you're moving through the Golgi apparatus, that membrane is gradually getting thicker and thicker from the cis-Golgi network starting here to the trans-Golgi network, which ends here. So proteins can move between the different compartments of that Golgi apparatus until the membrane thickness is too big. And once the membrane thickness is too big for the transmembrane domains, that stops that protein from moving on. We can target lysosomal proteins, both to endosomes and to lysosomes, to be involved in breakdown of materials. Within the ER, there are soluble lysosomal enzymes that will be N-glycosylated, and those glucose and mannose units can be removed. Within the Golgi apparatus, the mannose will be phosphorylated by 2 Golgi-specific enzymes. One is within the early compartment of the Golgi stack, where phosphotransferase adds a GLC-NAC1-phosphate, and one is around the mid-Golgi, where a second enzyme removes the GLC-NAC and a mannose 6-phosphate remains. 3, within the trans-Golgi network, the mannose 6-phosphate tagged enzymes will bind to the mannose 6-phosphate receptors. That whole thing will be packaged into a vesicle and taken to an early endosome. Then the early endosome will mature into a late endosome, and the pH will eventually decrease and cause the receptor and enzyme to separate. Then the receptors are recycled in vesicles back to the trans-Golgi network, and the late endosome either matures into a lysosome or delivers its contents to an active lysosome. Degrading and recycling any components that are unneeded or damaged can be carried out by specialized multivesicular endosomes, or MVEs. Sometimes they're also called MVBs for multivesicular bodies. These multivesicular endosomes are an intermediate between early endosomes and lysosomes, and they sequester the materials that will be destined for degrading and recycling. There are stop transfer sequences that help make sure integral membrane proteins get integrated into the membrane correctly. So that polypeptide of an integral membrane protein will be synthesized just like any other protein, any other soluble protein. But then that completed polypeptide chain will remain in the ER. And there's 2 different mechanisms that will help anchor that polypeptide chain within the bilayer of the ER membrane. The first mechanism is using polypeptides with an ER signal sequence at their N-terminus. With this, the SRP can bind to the ribosome and take it to the ER membrane. The elongation of the polypeptide will continue until that hydrophobic transmembrane domain is synthesized. And that hydrophobic amino acids, they function as a stop transfer sequence that stops the translocation of the polypeptide through the membrane of the ER. And as translation continues, the rest of the polypeptide will stay in the cytosolic side of the ER. So here you can see this. It starts being created on the cytosolic side. And that stop sequence will move laterally and form this permanent transmembrane segment. The second mechanism is using membrane proteins without a typical sequence at their N-terminus. They have an internal start transfer sequence that the SRP binds to and targets it to the ER membrane. And then its hydrophobic region just acts as an ER, as a transmembrane anchor. A lot of important membrane receptor proteins are 7 membrane proteins. They have 7 membrane spanning regions. And to do this, you have that same basic sequence of a start transfer that puts it in and a stop transfer that lets it go back out. Then a start transfer starts this, stop transfer lets it go back out, start transfer, then stop transfer. So that's just going to gradually thread these different regions within the membrane of the ER. And that process continues until you've made all of your transmembrane sequences and properly inserted them into the endoplasmic reticulum membrane. Once that polypeptide is incorporated into the membrane of the ER, it can stay there and work as a membrane protein within the ER, or it can be transported out to the Golgi or to other components of the endomembrane system. And transporting it to the appropriate compartment is going to be carried out by membrane budding and fusion of those vesicles. There's also post-translational import. That is another way to import things into the lumen of the ER. So if a protein is made on a ribosome within the cytoplasm, it can subsequently be shifted into the ER through post-translational import, similar to how co-translational import was happening. And as this protein is going to be made, it associates with HSP70 chaperones, and that keeps a protein unfolded, as you see in this image. That complex of the ER membrane proteins will target the membrane protein for translocation, and the protein moves through the pore, and as it does that, it loses these chaperone proteins. And as it gets into the cell, BIP, this little protein BIP, is going to be recruited here where the protein is coming through. And BIP uses ATP hydrolysis to help pull the polypeptide into the lumen. All right, so we've talked about different ways that we target our protein to different regions of the cell. We're going to come back and talk a little bit about exocytosis and endocytosis, how we are kind of interacting across the plasma membrane in our next video. I'll see you soon. Bye.